Okay, welcome everyone. This is our lecture and note-taking on the topics of RNA and transcription. You should have your handout that was linked on the website so that you can take the notes as we move along. Let's go. All right. So, like I mentioned before, this whole unit is around the central dogma of biology, where DNA is transcribed into RNA, which is translated into protein. Previously, a couple days ago, we took notes on the idea of DNA replication, the way that DNA is copied when a cell undergoes mitosis, when a cell is growing. Today, we're going to talk about this step where DNA is transcribed. The process is called transcription into RNA. And we're going to talk a little bit about what RNA is and then how this transcription process works. Okay, so let's get going. So um, RNA is a nucleic acid, just like DNA is, but it's a little bit different. We're going to talk about some of those differences. So first of all, it's called RNA, ribonucleic acid, instead of deoxyribonucleic acid for DNA. Okay, so there's one of the differences. The structure, the basic structure of RNA is similar because it's nucleic acid just like DNA is. So it is made up of nucleotide building blocks, just like DNA is made up of nucleotide building blocks. The difference, though, there are some differences between these building blocks. So let's investigate. First of all, there's still a phosphate group, just like there was before. There is a sugar molecule. There's a little bit of difference there. And there is a nitrogen-containing base, just like there was before with DNA. Let's talk a little bit about what these differences are. So, first of all, um, uh, with DNA, we had four bases, A, C, T, and G. With RNA, we have four bases, but uracil replaces thymine, so there is no T in an RNA molecule. There is a U instead. And the base pairing rules are like before, where like before, C pairs the G. But now, if there is a DNA molecule has an A, then your RNA molecule will have a U instead. All right? So if this is our DNA molecule, A, T, C, G, A, C, A, T, T, C, G, what do you think the corresponding RNA molecule would look like? What would that polymer be? Remember, use the same base pairing rules. So take a second, write down what you think the corresponding base pairing bases would be in an RNA molecule for this DNA molecule. Okay, did you get this? When there's an A in DNA, there's a U corresponding in RNA. When there's a C in RNA and DNA, there's a G in RNA. When there's a T in DNA, there's an A in RNA. All right? So hopefully you got this. These are the base pairing rules for RNA. All right. Another difference between RNA and DNA is actually not another difference. The same difference, the one that we're talking about, is the difference between thymine and uracil. Thymine you find in DNA, uracil you find in RNA. If you look at these two structures, you see they're very much alike. The only difference is that where thymine has this CH3 group right here, uracil has nothing. That's the only difference between the thymine, which you find in DNA, and the uracil that you find in RNA. Now don't worry, I'm not going to ask you to draw these structures or even to recognize them. I'm just pointing out what the differences are. So one of the differences between RNA and DNA is the presence of uracil instead of thymine. Another difference between RNA and DNA is the sugar molecule that helps to make up the backbone of the nucleic acid. DNA uses a sugar called deoxyribose, and that's this structure here. RNA uses a very slightly different sugar called ribose. The difference between them is this right here. So ribose has an OH right here, an oxygen and a hydrogen. Deoxyribose only has a hydrogen. It's deoxy, no oxygen. That's the difference between them. Otherwise, these sugars are the same. All right. Another difference between RNA and DNA is that if you remember when we talked about RNA the other day, I'm sorry, when we talked about DNA the other day, DNA needs to be double-stranded. It needs to have two strands of DNA wrapped around each other in a double helix for the structure to be satisfied. DNA does not like to be single-stranded. RNA, on the other hand, is perfectly happy to be just one single strand of RNA. It doesn't need a partner strand. It's okay. It's stable without a partner, unlike DNA. DNA unstable, single-stranded. RNA is totally happy, single-stranded. All right. So, um, we only talked about when we talked about DNA, there was only one kind, just the DNA that you find in the nucleus. With RNA, there are actually three different types. So let's talk about the three types of RNA. The first one that we're going to talk about is called mRNA, and the M stands for messenger. All right. So messenger RNA is built from a DNA template. We're going to talk about that process in a couple of minutes. Like I said before, it's RNA, so it's happy to be single-stranded. Instead of thymine, there is uracil. And as it turns out, messenger RNA is also the largest type of RNA. The function is basically mRNA serves as the recipe for making a protein. So the DNA is like a cookbook, right? And in the cookbook are a bunch of recipes. Each of those recipes is like a gene, all right? I made this analogy before, but I'll make it again. You want to think about the nucleus as a library where you can't check out books. You can't if you want to write down one of the recipes that you find in the cookbook inside this nuclear library, you have to write it down. You can't take the book home. And that written down recipe is the messenger RNA. So messenger RNA's function is to carry the recipe, the genetic code, from the nucleus out to the cytoplasm where the ribosomes are so that you can make the protein that you need. So think of mRNA as your recipe. And so we're going to talk about this process. This is an animation of an RNA, a messenger RNA molecule being made inside the nucleus. So the blue strands are our double helix of DNA. You can see that they start out wrapped around each other. And then this large green egg-shaped thing at the beginning of the animation comes along. We're building this red strand of mRNA. And after we finish copying or transcribing this message, you can see that the red strand disconnects and goes away, and the DNA has rewound itself. We're going to talk about that whole process in a couple of minutes, but this is basically how you make a messenger RNA molecule. All right, the second type of RNA is called tRNA, and the T stands for transfer. So tRNA is transfer RNA. Some interesting facts about transfer RNA. It's a three-dimensionally shaped molecule. At one end of this molecule is an amino acid. Now, we haven't talked about this a whole lot, but when you build a protein is built from amino acids. So amino acids are the building blocks or the monomers for a protein chain. And the tRNA is going to help produce that protein chain, and it does so by having one specific amino acid at one end of a tRNA molecule, and at the other end of the tRNA molecule is a sequence of three RNA bases called an anticodon. And this anticodon will match up with three nucleotides on the message RNA. Again, we're going to talk more about translation in a couple days, but I have to introduce this now. So the function of tRNA is to deliver an amino acid to the ribosome so that the ribosome can make the protein that it's supposed to make. Okay? So tRNA is like a delivery vehicle, and it helps decode the recipe, the message that's in the messenger RNA. This is what that three-dimensional structure looks like. So there are a lot of different tRNAs. There's around 60 different tRNAs. And the reason for that will become apparent when we talk about protein translation in a couple days. For right now, just trust me, there are a bunch of different tRNAs. They all have the same basic shape. So we call this a flattened cloverleaf shape because of these loops right here, here, and here. At one end of this molecule will be attached a, a specific amino acid. And at the other end of this molecule um, is a sequence of three nucleotides. They're shown in red here. Three nucleotides that make up what we call the anticodon. And this anticodon becomes important in the translation of the message that we'll talk about in a couple days. All right. So here, this little cartoon is to help us try to get some understanding around that. So here's our messenger RNA. This is the copy of the recipe that came from the DNA inside the nucleus. And so that's our mRNA. And and there are three nucleotides. So the way you read a messenger RNA is to read three nucleotides at a time. That's what makes up a codon. So three nucleotides, 1, 2, 3. These three U's are one codon in the messenger RNA. And right before that, there was C, C, C. That's another codon. Before that was A, G, G. So you're reading this three nucleotides at a time. Again, we'll come back to this during the translation piece. But here is our transfer RNA molecule, tRNA. And you can see it has this distinctive shape. At one end is an amino acid, and at the other end is an anticodon. Now, like I said a couple minutes ago, there are a whole bunch of different transfer RNAs, and they serve to map amino acids to anticodons. So this anticodon, AAA, which will match this codon, UUU, when this anticodon, AAA, always has the amino acid phenylalanine attached to the other end of that tRNA molecule. So if your anticodon is AAA, then your amino acid is phenylalanine. If you had a different anticodon here, you would have a different amino acid here. All right. Okay, again, we'll come back to the translation details later, but just try to get us started. Okay, and this matching up right here where the anticodon recognizes the codon on the mRNA, this is will be happening inside the ribosome. All right. All right. The third type of messenger RNA we're going to talk about is called rRNA, or ribosomal RNA. Now, we've talked about ribosomes before. They're organelles that you find in the cytoplasm of the cell. We call them protein factories because they make proteins. We now know that the recipe or the message for that protein comes from messenger RNA. But we haven't actually asked the question, well, what is a ribosome made of? And it turns out ribosomes are made up of RNA. In fact, they're made up of ribosomal RNA. A ribosome has two pieces, two subunits. There's a small subunit and a large subunit. And those subunits are made up of ribosomal RNA strands. All right, and that's what makes up a ribosome. All right, so here's a cartoon that shows us that. So this brown object is our ribosome. It has a small subunit here and a large subunit down here. And here is the messenger RNA molecule that's going to be read, going to be translated, sandwiched between the small subunit and the large subunit. All right. This is this is what we'll come back to in a couple of days when we talk about protein translation and how this process works. Okay, to summarize that, we have three types of RNA. There's messenger RNA, that's the recipe for a protein. There's transfer RNA, that's the kind of RNA that delivers amino acids to the ribosome to build up the protein. And then there's ribosomal RNA, which is like the structure of the ribosome itself. All right, let's talk a little bit about, well, the rest of the time we'll talk about transcription. That's the process of actually making an mRNA molecule, which is a copy of a DNA gene. So your cell needs to make a given protein. The recipe for that protein is in the DNA. But of course, DNA doesn't leave the nucleus, so in order to use that recipe, we need to make a copy of the recipe. That's what mRNA does. Transcription is the process of making that copy. Come on. Okay, quick comparison of RNA transcription and DNA replication. Kind of hit a couple of these points before, but here they are again. RNA is single-stranded. DNA does not like to be single-stranded. Has to be double-stranded. RNA is single-stranded. There's uracil instead of thymine in RNA. And then something we haven't mentioned before is that RNAs only transcribe. You only make a copy of a little piece of the DNA chromosome because DNA is huge, right? You have like 3 billion DNA bases inside a cell. You don't want to transcribe or make a copy of all of that at once. You just need one particular gene, the recipe for one particular protein. So RNA transcription takes place only in little tiny pieces only at certain times, whereas DNA replication, you're copying the entire chromosome, all the DNA in the cell, all at the same time. So RNA transcription only happens in small pieces, whereas DNA replication happens all at once. Okay, we had 5 steps for DNA replication. We have 6 steps for RNA transcription. Let's talk about those. The first one, they're going to look very similar. All right, so pay attention. The first one, helicase and primase will untwist the DNA and start the process. That should sound familiar. The DNA unzips. That's step 2. Now before, we called this a replication fork. Now it's called a transcription bubble. It's kind of to help us keep track of what's actually going on. The third step is for free nucleotides to base pair with the DNA. But of course, these are RNA nucleotides, not DNA nucleotides, because we want to make an RNA strand, not a DNA strand. A pairs with U and C pairs with G, like we talked about earlier. So here's a little cartoon. This is not a very good picture, sadly. So there's a better one on the next slide. I think that one's better. So let me move my picture. So the fourth step is for the RNA nucleotides, the free RNA nucleotides that are kind of floating around inside the nucleus, will base pair with the exposed DNA backbone right there. The exposed DNA strand. And you can see in this picture, here are the free nucleotides. They will base pair with corresponding DNA nucleotides on this strand. And then this enzyme, RNA polymerase, will connect the nucleotides to one another, just like DNA polymerase did the same job, only for DNA-free nucleotides. After you've completed the entire message, the new single-stranded RNA will detach itself, and the DNA will close up, rezip itself, retwist itself, and the process of transcription is complete. What we end up with is a single-stranded mRNA, which just corresponds to the one gene that you need a protein for. And the mRNA will leave the nucleus and go to the cytoplasm to find a ribosome so that it can be translated into a protein. The DNA, of course, stays in the nucleus. It is unchanged by this process. It's just being copied. Good? All right, so here's our little diagram again. So this big green egg-shaped object is your RNA polymerase, and you'll notice that first the DNA untwists. That's helicase and primase. We're not showing those here. And then RNA nucleotides, which are these red backbone, are being base paired and added one by one, and the polymerase, the green object, is connecting the new nucleotides to one another. And when that's done, the red strand, single-stranded, disconnects from here, from the DNA, and it leaves, and the DNA goes back to its normal double helix, and the process is over. All right, so That brings us to last slide here, I think. Our central dogma again. What we've been talking about here is the process of transcription, where you make an RNA copy of a DNA molecule. This happens inside the nucleus, um, and it happens when you need to make a particular copy of a particular gene so that you can make its protein. We'll talk about this translation step in another lecture. All right, that's it.